Assalamu alaikum to you all. Welcome in Way of Knowledge. In this video, we will solve and understand the compulsory reasoning questions of biochemistry from Genetics paper 4. Reasoning question number 1. DNA polymerase is inactive if beta subunit is absent. Reason is that because it acts as sliding clamp that catalyzed the synthesis of leading and lagging strand by binding with DNA polymerase holoenzyme. Number 2 is lactoperon is replaced in the presence of low CMP level because CMP, cyclic AMP, binds to cap forming a complex that enhances the transcription of lactoperon. Presence of glucose lowers intracellular cyclic AMP level, so transcription of lactoperon is replaced in the presence of low cyclic AMP level. Number 3 is transcription of DNA by RNA polymerase requires the participation of topoisomerase 1. Reason is that transcription of DNA by RNA polymerase holoenzyme requires the participation of topoisomerase 1 to release the transcription generated helical stress after the formation of transcription bubble. Sigma factor is important for transcription. Answer is that because sigma factor initiates transcription by binding with RNA polymerase to direct the addition of new ribonucleotide. Number 5 is DNA replication essentially requires DNA polymerase 1 because DNA polymerase fills gap after removal of primers. Number 6 is post-translational modification is of its significance. Reason is that because it processes mRNA for protein synthesis by removing intron non-coding sequence of protein or amino acid and joining exon according sequence of protein and amino acid. It also adds 5' cap of methyl guanosine and polyadenylate tal. Number 7 is 32 transfer RNA are sufficient for 61 codons. Reason is that because anticodon of each transfer RNA interacts with more than one codon. Number 8 is a primer is required to initiate DNA synthesis because DNA polymerase can add new nucleotide with 3' OH of nucleotide. Number 9. Single-stranded DNA binding protein is important for DNA replication. Reason is that because it prevents recoiling of DNA strands by binding with single DNA strand. And last but not the least, reasoning question number 10 is a specific sequence on messenger RNA directs its positioning on ribosome. Reason is that because this sequence, which is called Shine-Delgado sequence, helps in the initiation of protein synthesis by annealing the ribosome with starting codon AUG. So I hope you will get and understand the answers of these reasoning questions. Thanks for watching. Till then, Allah Hafiz. We will meet soon in the incoming videos.